Right, I hope you all had a lovely lunch and a cup of tea or coffee to finish off so that we can enjoy the last session of this wonderful meeting where we saw some great science and fantastic interactions. And I must not to make jokes about the session, okay? So I'm going to simply read it without saying anything. Microglial sex and aging. And Marina is going to open the session from Trinity College, Dublin, introducing the topic. Thank you.
Thank you very much for the invitation here today. I'm delighted to be here. And I hope that I'm not going to mess up the slides because I normally do. Okay, so first thing I want to do is to point out to you something that you already know, which is that about 2/3 of people with Alzheimer's disease are women. And most of the evidence would suggest that there are differences in the presentation and differences in the progression. Possibly, there's a bit of a dispute about the difference in the amount of pathology that's found in human female AD patients compared with males, but in animal models it seems to be fairly clear that the females have more pathology. Now, this slide shows something that most of you are very well familiar with, and that is that there's a sequence of events that appears to occur, beginning with the laying down of amyloid, followed by changes in brain structure, memory, and then clinical function. But recently, the evidence is pointing towards a role for inflammation and consequently, potentially, microglial changes in earlier stages of the disease, and also changes in metabolism. And I'm going to be talking about both of these here today. So you're very well aware of all these beautiful papers indicating that changes take place in microglia in either the human brain or in Alzheimer model brains. And what I'm going to do is just report what we have found, first of all, in APP-PS1 mice, the model of Alzheimer's that we used. And it started several years ago when a PhD student in the lab took bunches of 4 bunches of animals, wild-type males, APP-PS1 males, wild-type females, APP-PS females, and these were 18 to 21 months of age. And right the way through, I'm going to be putting the males first and the better wine at the last, there, the females in red. So he just simply looked at a whole range of cytokines at messenger RNA level and the secretion of these in microglia that were prepared from these 4 bunches of animals. And when he split them into the 2 sexes, what he found was there were little changes in the males in TNF-alpha, IL-6, and IL-1, but the changes were much more evident in female APP-PS1 mice compared with female wild types or male APP-PS1 mice. So that was the beginning of something that persuaded us it might be worth looking a little bit further at the sex differences that occur in isolated microglia from these groups of animals. Now, I should say that some of these studies were part of an original study where we looked at young mice, both wild-type and APP-PS1, males and females, and we found no differences. So I'm just not referring to those here at all. So now, the first thing that we did was a nanostring analysis. This is a good while ago, you'll appreciate. And with that, what we found was, if you look at this heat map, it's very telling, I think. The males, wild-type and APP-PS1s, are the first 2 panels, and then the females are the second. And the significant thing is that there's a lot more upregulation of genes that are associated with microglial function in the female APP-PS1 mice compared with any of the other groups. Some small changes in males, but much more evident in females. Now, I've just picked out 5 of these, because these are 5 that you're very familiar with, Axel, CTS7, etc., etc. And what you can see is that in each of these cases, the change in female APP-PS1 mice is much greater than the change in wild-type mice or compared with male APP-PS1 mice. And here's a volcano plot showing more or less the same thing. Okay, now this, it's almost an insult to show you this, but anyway, I'm going to, because I want to draw your attention to something that I'm going to talk about. So the microglia are very busy cells. That's what I say at the top, and that's the most important message. So you can see they're moving all the time. Whether there is or not a response to injury, it doesn't make any difference. So the processes are all the time moving. The cell bodies actually move in response to a stimulus, and therefore they require a huge amount of energy. So the whole idea, of course, is to eliminate any danger that's there, and so as well as becoming motile, the cells sometimes are secretory and also phagocytic. And this tells us something about them. They absolutely must have a good supply of ATP. So what we did next was we took the same groups of mice and we looked at metabolism. So again, at the risk of boring you and insulting you, I just want to point out that glucose is broken down first by glycolysis to pyruvate, and with that set of reactions, you get 2 miserable ATP molecules. Now, if on the other hand, the breakdown continues to completion, where you get CO2 and water by means of mitochondrial metabolism or oxidative metabolism, then you get 32 to 36 ATP molecules, much better value, in other words. So we investigated ECAR, which is a means of assessing glycolysis in these isolated microglia from the 4 groups of mice that I mentioned. And what you can see, first of all, is that the 3 groups that I have shown here at the moment, the wild-type males, APP-PS1 males, and wild-type females, they all show much the same glycolytic profile. But when you add in the APP-PS1 females, what you see is that there's a much greater glycolysis going on in these mice. And when you do when you prepare histograms, you can see that this is also the case with the APP-PS1 mice. What's that? Jeepers! Do you know what? I can never. Oh, it's only on my screen. That's all right. So, okay. I need to get rid of it all the same. It's very telling stuff, not pornography now or anything, but... Oh, I don't know what to do. If I escape... No. Okay, I'm going to have to turn to this here. So when you turn this into histograms, what you see is the APP-PS1 mice have a much higher signal, and this is glycolytic signal, than the males or the other groups. And in addition to that, you have greater lactate acid in these mice compared with the males. Now, this is a paltry change, but nevertheless, this is what we found. My message here is that the microglial cells have different female microglial cells from APP-PS1 mice have a different metabolic profile. They seem to rely more on glycolysis than the males or wild-type mice. So, next. My colleague and daughter in the Harvard Medical School was looking at metabolomics by mass spec, LC and mass spec. And so we sent her cells, and because she owes me an awful lot, she did the analysis for us. And again, I have to just point out that over on the far, the heat map shows the same profiles. Again, the males in the first 2 and the females in the second 2 panels. And what you see are differences which are identified in red that seem to be associated with the metabolites that are linked with glycolysis. And a few of them I have highlighted here. You will see that some do show sex-related changes as well as genotype-related changes, but not all. And what I wanted to do was point out that we got a nice proxy marker from several experiments that we did that is indicative of a glycolytic cell, and that is the presence of PFK-FP3, this enzyme, in the cytosol. So it translocates from the cytosol from the nucleus to the cytosol when the enzyme is active. And when it's active, it drives this reaction fructose 6-phosphate to fructose 1,6 bisphosphate, and is actually a very key controller of glycolysis. So when we looked by staining in these cells from male and female APP-PS1 mice, what we found was that there was a decrease in PFK-FP3 staining in the nucleus of female APP-PS1 mice, accompanied by an increase in cytosol, which indicates to us again that these cells are glycolytic. Now, one thing that I'm going to point out is that at the top of that heat map, you see a number of metabolites which are downregulated in female APP-PS1 mice. And I'm picking out two because they're two metabolites which are linked with the oxidative metabolism, and I'm going to come back to that in a second. But one thing that we found out about these PFK-FP3 positive cells, those are the cells with the PFK-FP3 in the cytosol, was that they seem to cluster around A-beta plaques. So the A-beta plaque is shown here in green, and then round about those you can see a collection of cells that appear to have PFK-FP3 in the cytosol, suggesting to us that the cells that are closest to the plaques might well be glycolytic cells. And in contrast to that, we don't see very many of those cells at all, distal from the plaque. Now, what that means is a mystery. So I mentioned to you already that oxidative changes were suggested by the metabolomics, and here we see that yes, indeed, we do have changes, but these changes are a little bit weird. So again, we use Seahorse to look at OCR. This is the oxidative metabolism now, and if we just concentrate on the histogram, it might be a little easier. But what it seems to show us is that microglia from APP male mice are able to upregulate oxidative metabolism, whereas females don't seem to be able to do that. And this is one of the signs, I think, that we have a different type of plasticity in the cells in males and females. And we looked to try and link that with some sort of function, and we investigated phagocytosis and also chemotaxis and motility, but I'm just showing you the phagocytosis here. And what we saw was the phagocytic ability of female APP-PS1 microglia was reduced compared with the males. And that was associated with an increase in plaque area and plaque number. These differences are not huge, but they're there. And in addition to that, we saw other changes that are sex-related. And again, I'm just going to ask you to look at the histograms because you'd be demented if you had to look at all these things. But what we looked at were indicators that cells might not be as healthy as they should be. So the two markers that we looked at were P16 and lipofuscin. And as you can see, we get genotype-related changes in both of these, but also significantly sex-related differences. And once again, the differences seem to be more profound in the females. Now, I've got a number of slides showing you that morphology changes. And so here are the first few bits of information. So first of all, when we looked at the actual shape of the cells, what we found was far more rod-shaped microglia in female APP-PS1 mice compared with males. Really an awful lot more. And then when we did further analysis, we found something interesting again that I think might reflect this plasticity issue that I have, because it looked like the male wild-type mice, which were similar enough to the female wild-type mice, had plenty of processes and so on, but these were able to convert into a much more amoeboid-looking cell compared with the female APP-PS1 mice, which seemed to still retain their processes. Now, What that means, I don't know, because we don't like talking about function related to morphology with very good reason. And consistent with this, when we looked at various morphological parameters, we saw that the number of branches and the number of junctions and lots of other things were different in aged APP female compared with aged APP males. And you can see plainly there that there's a very significant difference. The next thing we looked at was iron accumulation in the cells, and we found that the iron accumulation in female APP-PS1 mice was far greater than in males, and the iron-related cells seem to cluster around the A-beta plaques. Now, we don't know yet whether these iron-related cells are glycolytic, but what we do know is that when cells accumulate iron, that is when we incubate them in the presence of ferric chloride, as you can see up here, that the cells do become more glycolytic compared with the controls. So iron accumulation increases glycolysis. It also increases inflammatory cytokines, and here I'm just showing you TNF, and increases, and it is with the greatest of trepidation that I say the term microglial activation. Okay, so what do we find? We see something of a cycle, and we're not really sure yet how to break the cycle down, but we know that inflammatory changes in the brain are linked with microglial cells that are glycolytic and also iron positive, and that this combination seems to lead to a compromise in the cell's energy, such that these cells switch towards a glycolytic phenotype, and the cell function deteriorates. And I showed you the phagocytic information, and amyloid increases. In addition to that, the cells change their shape, and to make a longish story short, the male cells in the APP-PS1 mice become amoeboid, but the females don't, and in fact, they're rod-shaped. So we then decided we'd have a look at samples that we got from the Netherlands brain banks from males and females that had died and donated their brains. These people all had similar BRAC scores, similar ages, and so on. And so, as you see, we only had a small number of samples because that's all they could match for us with males and females. So we had 5 male samples from AD patients and 4 from age-matched controls. So we looked at what we could look at, given the tissue that we had, and the first thing was the morphological differences. And you can see here in this picture, the top panel there, that there's a very big difference in the morphology of the microglia taken from AD male patients compared with controls. And the difference isn't nearly as marked in the females, as you will see. And when we did some other types of analysis to try and tease this out a little bit, what we found was that many of the morphological markers that we identified in mice were present here. In particular, we found that in AD, the males, the cells became amoeboid, and this didn't seem to happen nearly so much in the females. Indeed, what we got were plenty of these rod-shaped microglia, just as we found in the mice. And again, the change was much more profound in females compared with males. And we also found these funny-looking microglia that we're calling dystrophic, because it seems like a nice word. And you can see that the processes are broken up, and they're all gammy-looking cells. So dystrophic microglia in females compared with males. In addition to that, we found that iron accumulation occurred in the microglia in females to a much greater extent than in males, and that's clearly evident here. Here are the 2 females. You can see very different changes in these. And in addition to that, if you look down at the very bottom of this slide, you'll see that the iron seems to accumulate all around the cell, also at processes, at the very ends of processes. So we have these little spheroid things going on. So here we have a genotype-related change in the iron accumulation in the cells, which is greater in female AD patients compared with males. And we also looked at the 2 markers of cell health, I suppose, that we used in our mice. And once again, we see more P16 positive cells in the tissue from Alzheimer female patients compared with males, and the same applies to the lipofuscin. Now, this is, in a sense, you need a little bit of faith to look at the lipofuscin, but they're kind of darker, purpley-ish brown instead of just plain brown, for the want of a better description. And finally, we looked at the A-beta plaques in male and female AD patients. And what you'll see is that there's some evidence of a slightly greater A-beta plaque area and also number. And these are 2 significant changes, but, they're not wonderful, are they? Anyway, that's what we found. And an interesting thing is that there was far more A-beta accumulated in the vasculature of males compared with females. And I hope you can see that there. Lots more, and you can see the graph illustrating that. So my conclusion. So with the changes that occur in both our animals and our human samples, we see that females seem to have more profound changes than males. We see that inflammatory changes seem to be greater in females compared with males. Metabolism in the mice, we tried to stain for PFKFB3 in the human tissue, but it didn't work very well, and so I'd rather not show it to you, but if you were an optimistic person, which I am, you'd be inclined to think there was greater staining in females, but we're never going to say that in public. Okay, so the sex-related differences are in metabolism, in function, in inflammation, in morphology, and in iron retention. And of course, as usual, we're left with more questions than with answers. So, for example, the microglia in females, they're definitely dystrophic. Does that mean anything? Does it mean they're dysfunctional? They're rod shape. Does that mean that they're persistently activated? We don't know. They're iron retentive. Does that mean that that links up with our mitochondrial change, the oxidative change? And we don't know that either. And of course, the biggest problem is we have no idea which of these changes, whether it's metabolism or inflammation, kick-starts the whole thing. But what we do know, and what I can be absolutely firm about, is that females are much worse off than males. And do you know what? Isn't that the story of our lives? Okay. And an awful lot of people contributed to this, and so I've those highlighted in black that really had a very big part in the talk that I've given here. And I have to make reference to my collaborators in Trinity, and especially well, why especially us? Because she's my daughter, in Harvard Medical School. Thanks very much.
Thank you so much, Marina. Great talk and fantastic entertainment as well. Questions, please. Jake, would you like to start? Oh, sorry.
Oh, great. Thank you so much for your presentation. That was really fantastic. Obviously, do you know about the particular genetics behind this? Like what particular genes might be really influencing this? Because obviously, for me, it's just simply the lack of a second X chromosome or the Y chromosome in the microglia. Do you know about anything about the genetics?
The quick answer is I don't.
Okay.
But obviously there are a few things that can contribute to the differences, and you picked on genetics clearly. We have, that's one thing, chromosomal genetic thing. Second, we have hormones. And then the thing that I hate most of all, but which people insist might be important, are epigenetics, because I just don't want to go there. But it would be terrific if somebody with the transcriptional prowess went after this. I think it would be lovely to see.
Cool, thank you.
There was a question. Yep.
Apologies if I missed this, but do you observe gender-related differences in the proliferation of microglia?
Well, no, I didn't show anything about that, but I can tell you no is the answer, because we did check that.
Okay, thank you.
Yeah.
Thank you.
Another question here? Oh, yeah, please.
So thank you for the interesting talk. Does that
Oh, finally, sorry.
Marina.
does that lactate hypothesis of astrocytes secreting lactate taken up by neurons cause something different here? Might be more lactate in the presence microglia get more lactate present and inhibit the function, or microglia are producing lactate to support dying neurons. Do you?
Yeah, that's possible, but you know well, you made reference to it there, I think, that generally the idea would be that the lactate comes from astrocytes. Now, we looked at astrocytes. In the very beginning of this, we compared the changes that might take place in astrocytes compared with microglia. And there absolutely nothing happened in astrocytes in terms of sex-related differences, nothing. So we had no metabolic change whatsoever. So that suggests to us that you know, referring to the possibility that you just made reference, that astrocytes are still capable of providing the lactate to the neurons, and therefore, you know, why bother the microglia with that function as well as all the other things they have to do, you know? So the astrocytes in the experiments that we did on metabolism show no change, none.
Thank you.
Maybe I can go. Thanks very much. That was a really excellent talk. I'm wondering if the rod-shaped microglia might be lining blood vessels.
It's possible. We did not look at that, and we could easily do so. So thank you.
I'll sit down. Yeah, hi. Thanks, Marina. That was very entertaining. Did you ever look at things like glutathione levels?
No, not in this. Not in this. We did years and years ago, but not in this.
There's quite a few recent studies where they put cells into the brains of animals, microglia. I just wondered if you were aware of any studies that had compared whether you put female or male microglia. So is it a cell autonomous change or an environment that's influencing?
Well, it'd be just extraordinary if the changes, that sex-related changes, were confined to microglia. But I don't know, I can't answer that question. I just simply don't know. But if you look at the literature now and ask whoever, Google or whoever, to tell you about how many papers have been published on sex and microglia in recent times, you'll find that the number is increasing exponentially. But if you look at other cells, not so much at the moment. So I'm afraid I can't answer that question. I'm a dab hand at not answering questions. I'm sorry.
Anybody else, please? Perhaps. Oh, I thought there was a hand. Marina, perhaps. I wonder if there is any evolutionary, if there are any evolutionary changes to mitochondria depending on sex. So, you know, males versus females, you know, early exposure to hormones or something like that.
Probably is the answer to that, because for sure there are people that look at the developmental changes very early on in P5 and so on, and they do see differences in metabolism between males and females, and indeed in an awful lot of other things apart from metabolism. But I'm unaware of anyone that tracked changes through the lifetime. Just seems that a lot of those changes during the developmental period sort of settled down, and there aren't huge sex-related differences in, let's say, inflammatory markers or cell function from maybe 28 days until much later on. I mean, we got some changes at 9 months, but I didn't show you those because it's hard at 9 months. Some animals look like they've aged. Some look like they're spring chickens. So, yeah.
Thank you very much.
Thank you.